In a laboratory in New York, a pathologist bends over a slide. He is Frederick Zugibe, Chief Medical Examiner of Rockland County, Adjunct Associate Professor of Pathology at Columbia. He does not know where the sample came from. He has not been told. That was deliberate. He looks. And then he says what he sees: heart muscle, left ventricle, inflamed, infiltrated with white blood cells that should not be there. This heart, he says, suffered a violent trauma to the chest. He is looking at a communion wafer. A 15-year-old boy in Milan would spend the last years of his short life gathering cases like this one. He found 187 of them. This is sign of contradiction. His name was Carlo Acutis, born in London, the 3rd of May, 1991. Raised in Milan, the son of a banker and a publisher, neither of whom practiced the faith when he began to. He was, by every account, an ordinary teenager. He played football. He owned a dog. He limited himself to 1 hour of video games a week. He was also, from childhood, at daily mass, and he could program computers. He taught himself, asking his mother for the textbooks trained engineers used. In October of 2006, at the age of 15, he died of acute promyelocytic leukemia. It took days. 19 years later, on the 7th of September 2025, Pope Leo XIV declared him a saint, the firstborn in the millennial generation. But this episode is not really about Carlo Acutis. It is about what he found, because before he died, he built something, and what is inside it is far stranger than the boy who assembled it. He built a database. Over roughly 2.5 years, Carlo compiled documented reports of Eucharistic miracles, cases in which a consecrated host was claimed to have bled or resisted decay or become something that was no longer bread. He photographed. He indexed. He cross-referenced dates, dioceses, and investigations. 187 cases. He turned it into a traveling exhibition that has since crossed 5 continents. Now understand what this means. This was not a boy collecting legends. Among those 187 entries are cases that were taken into forensic laboratories, subjected to electron microscopy, immunohistochemistry, and DNA analysis by pathologists, cardiologists, and forensic scientists on 4 continents. 3 of them stand above the rest for the depth of the analysis performed: Buenos Aires, Sokółka, Tixtla. 3 countries, 3 decades, 3 consecrated hosts, and, according to the laboratories, the same tissue. August 1996. The parish of Santa Maria in Buenos Aires. A consecrated host is found on the floor, soiled. Following the ordinary rule of the church, a priest, Father Alejandro Pezet, places it in water in the tabernacle to dissolve. It does not dissolve. Within days, red stains appear. They grow. The Archbishop is informed. His name is Jorge Mario Bergoglio. He will one day be Pope Francis. He orders the host photographed and left where it is. It stays in that water for 3 years. It never decomposes. In 1999, an investigation begins under Ricardo Castanon Gomez, a Bolivian scientist and former atheist. A sample is taken on camera under a documented chain of custody and flown to New York. It goes to Frederick Zugibe, and Zugibe is not told what it is. He examines it blind. His conclusion: cardiac muscle, myocardium, from the wall of the left ventricle near a valve. The tissue is inflamed and infiltrated with leukocytes, white blood cells, which do not belong in heart tissue unless the heart has been injured. The pattern of damage, he says, resembles what he has seen in men beaten severely across the chest. And then the detail that stopped him. The white blood cells were intact. In distilled water for 3 years, they should have been long destroyed. Their survival, he said, was what one would expect from a chemical preservative, not water. When they took that sample, he said, the heart was alive. The 12th of October 2008. Sokółka in northeastern Poland, the Church of St. Anthony of Padua. A consecrated host falls during the distribution of communion. Again, the ordinary rule, water, a vessel, dissolution. Again, it does not dissolve. A red mark forms at its center. Archbishop Edward Ozorowski sends a sample to 2 pathologists at the Medical University of Białystok. Maria Sobaniec-Lotowska, Stanislav Sulkowski, 30 years each in histopathological diagnostics. They work separately. Neither sees the other's results. They reach the same conclusion: cardiac muscle, human, and in their own published words, appearing as though taken from the heart of a living person in agony. Under the electron microscope, they identify cross striations. They identify intercalated discs, which exist only in cardiac tissue. They identify the central positioning of the cell nuclei within the fibers, a marker specific to heart muscle, and one that rules out a bacterial colony. But the finding that neither of them could account for was structural. The heart muscle was not sitting on the surface of the wafer. It was interwoven with it. Bread and myocardium fused at the cellular level, forming a single, continuous structure. Sulkowski noted something else. Left in water, then dried on cloth for years, without stabilization, without refrigeration, the tissue never underwent autolysis. It never self-digested. It simply did not decay. The 21st of October, 2006. St. Martin of Tours in the town of Tixtla, state of Guerrero. 600 people are on retreat. During communion, a religious sister turns to the priest beside her, weeping. The host in her hands is bleeding. Bishop Alejo Zavala Castro convenes a theological commission that same year. 3 years later, he brings in Castanon, the same investigator from Buenos Aires. The scientific work runs from 2009 to 2012 across laboratories in Bolivia, Guatemala, and the United States under blinded conditions. The findings presented in 2013. The red substance is blood, human hemoglobin, human DNA. 2 independent forensic teams, using different methods, conclude the blood did not come from outside the host. It emerged from within it. The blood type is AB. The outer layer of blood is coagulated, consistent with 2006. The layer beneath it, sampled in February of 2010, is fresh, intact white blood cells, red blood cells, macrophages actively engulfing lipids, a process that occurs only in tissue that is alive. And the immunohistochemistry returns myocardium, heart muscle, lacerated and showing the mechanisms by which a living body repairs itself. On the 12th of October 2013, the anniversary of Carlo Acutis' death, the bishop signed a pastoral letter declaring the events supernatural, a divine sign. Now step back and look at what the laboratories returned. Lanciano in the 8th century, Buenos Aires 1996, Tixtla 2006, Sokółka 2008. 4 hosts, 4 countries, 12 centuries between the first and the last. Every one of them, cardiac tissue. Every one, the left ventricle. Every one, signs of trauma and agony. And where blood typing was possible, Lanciano, Buenos Aires, Tixtla, the same answer. Type AB, the same blood group identified on the Shroud of Turin. No one at Sokulka knew what the Buenos Aires laboratory had found. No one at Buenos Aires had read the reports from Tixtla. Independent teams, separated by decades and languages, returning the same organ, the same chamber, the same suffering. Now, this channel does not flinch. so let us stand on honest ground. Rome has not ratified Tixtla. The bishop of the diocese has declared it. The Holy See continues to review it, as it does with every case of global significance. A local declaration is not a universal one. At Sokulka, the head of the university department publicly stated the sample had been tested outside official channels. The findings were not the department's, and he said so. The scientists Kelly Kearse and Frank Ligage have argued that these investigations lack standardized protocols and parallel control samples. These objections are real. They are procedural. None of them explains why heart muscle is interwoven with bread. None of them explains why the cells were still alive. Carlo Acutis never entered a laboratory. He gathered the reports, he built the pages, and he died at 15 in a hospital in Monza, north of Milan, of a cancer that took him in days, years before the Tixtla findings would be published, years before the Sokółka professors would finish their work. He never saw the results. But the church that would one day canonize him required 2 miracles of its own, and it found them. A Brazilian boy in Campo Grande, born with a pancreas malformed around his intestine, healed after touching a relic, on the 12th of October 2013, the anniversary of Carlo's death. The very day in Mexico that a bishop signed the letter declaring Tixtla a divine sign. And a young woman in Florence, dying of a brain hemorrhage, restored on the day her mother knelt at his tomb. He spent his life pointing at the Eucharist. The Eucharist, it seems, pointed back.